So thank you very much for joining this webinar on my topic, recent regulatory updates on nitrosamines, NDSRIs from Malaysia regulations and European Medicines Agency regulations and FDA. Before I start my session, I would like to thank MOPI for giving this opportunity to present my understanding and my knowledge on this area, in this area. And I would also like to thank Ms. Leong. She is a consultant from Malaysia who has introduced me to MOPI. Thank you. Thank you, Leah. This is the disclaimer. So in the next 45 minutes to 1 hour, what I am going to present is basically I am going to talk about brief introduction on the nitrosamine impurities, NDSRIs, what are the recent recalls, what is the impact to the market because of this issue in last few years, and I am going to talk about the recent regulatory guidances on the nitrosamines. So in June 2023, Malaysia National Pharmaceutical Regulatory Authority has given some guidance on the nitrosamine impurities. I am going to brief about it. What are the brief guidance and what are the key takeaways from the guidance? Then there are very, very important updates from European Medicines Agency, EMA, in the month of July, dated July 7th and 28th. I am going to talk about that. That is revision 16 and revision 17. Then there is a update from FDA on the recommended acceptable intake limits for NDSRIs, guidance for the industry. This guidance has come on 4th August 2023. I am going to talk about it very briefly. I am going to also cover briefly what are the expectations around the analytical methods development, what is the sensitivity of the methods required, and then last, control strategy, and what are the challenges for controlling the NDSRIs. Then conclusions and way forward. So if you see the history of the nitrosamines, so the history is not very new. The history of nitrosamines, in fact, the nitrosamines produced in liver tumors in rats have been identified by British scientists in 1956. And there were about 300 nitrosamine impurities were tested and found 90% of them were carcinogenic. Then nitrate and nitrosamine intake are associated with the risk of gastric cancer and esophageal cancer. And again in 1970s also, the liver cancer was found in Norwegian farm animals. So it's not a very new identification of these impurities, but the history is quite old. And of course, nitrosamines impurities are present in water, animals, grilled foods such as bakery items, and drinking alcoholic beverages like beer and vegetables. So as long as the impurities are well within the exposure limits, it represents the low risk of health problems. But however, the exposure levels are above acceptable levels, over a period of long period, it may increase the risk of cancer. If you see about the recalls, almost if you can see this slide, there were about more than 1400 product lots have been recalled from the market due to nitrosamine impurities or N-nitrosamine impurities, what we call as NDSRIs, because of their presence beyond the acceptable intake limit. You can see a whole list of products, right from metformin, ranitidine story. You may are all aware of this. Ranitidine is almost, the drug is dead in the market. And you can see have hypertensive drugs like valsartan, amlodipine, losartan and all. So all these drugs, there were some recalls due to higher level of nitrosamine impurities from different companies. So what is the basis for the nitrosamine impurities? If you can see this slide, the basis is clear. I think nitrosamine impurities are the organic substances formed by the reaction of the secondary or tertiary amines with nitrate or other nitrosating agents. So you can see here the generic structure of dialkylamine when it having the reaction with nitrous acid, that is sodium nitrate plus HCl nitrous acid, it can give N-nitrazodialkylamine. So as I mentioned, these nitrosamine impurities are probable human carcinogens. As per ICHM7, they are referred as cohort of concern. So to form the nitrosamines, three things are required. As I mentioned, required amine substrate, then acidic conditions, and nitrate source. So these three things are required for the formation of the nitrosamine impurities. And very recently, there is a lot of focus from the regulatory bodies on the nitrosamine drug substance impurities, that is NDSRIs. So basically, if you see this slide, NDSRIs are the nitrosamine drug substance related impurities. If the API itself contains a secondary amine or tertiary amines and capable of nitrosating under suitable conditions. So if you can see here, the structure of dabigatran etixylate, you can see here the N-nitroso group. So if you can take out this NO group and if you have hydrogen here, it is a dabigatran. So the hydrogen is replaced with NO, that is N-nitroso group. So this is the this is called as NDSRI, that is nitrosamine drug substance related impurity. So basically, the everything API moiety is same. Only the NH group it is replaced with NNO. So these impurities are of late taking lot of importance and lot of work is going on in industry. So people also call these NDSRIs as complex nitrosamine impurities or API-like nitrosamine impurities or novel nitrosamine impurities or novel nitrosamines. So just to briefly understand on the what are the difference between the GTI, nitrosamine impurity, and NDSRI. So industry is working, since several years on the identification and quantification of genotoxic impurities in the API and drug products. So basically, when we work on the genotoxic impurities, we considered a TTC value of 1.5 microgram per day intake of genotoxic impurity associated with an acceptable risk, excess cancer risk of less than 1 in 100,000 over a lifetime for most pharmaceuticals. But when we talk about the nitrosamine impurity, unlike GTI, we only talk about specific acceptable intake value that shall be considered for arriving at a specification limit. So the acceptable intake value is generally with the unit of nanogram per day. So if I want to evaluate the specification limit for the nitrosamine impurity, I have to use a formula that the specification of the impurity, nitrosamine impurity, is equal to acceptable intake in nanogram per day divided by maximum daily dose in milligram per day. So that is the formula we use. So nanogram per day divided by milligram per day is ppm. And I already briefed about NDSRIs. So NDSRIs, as I mentioned, it is the class of nitrosamines very similar to the structural similar to API, but NDSRIs can be generated during the manufacturing or during the shelf life storage period of the drug product. If you see any regulatory guidance, so all the regulators, whether it is the European regulations, FDA, or even the recent guidance from the Malaysia, all the regulators has suggested a three-step approach for the nitrosamine impurities or NDSRIs. So there are three steps. One is the step one is the risk evaluation or risk assessment, what we call, and confirmatory testing, step two, and step three is changes to the marketing authorization. So basically, if I want to tell in a simple layman language, we have to identify the risk in the API or drug product. Then, if the risk is identified, we have to develop a suitable method. It is expected that the manufacturing authorization holder or product registration holder has to develop a suitable sensitive quantitative method, and identified nitrosamines need to be tested using a sensitive method. Then, if the impurities are beyond the limit of quantification in the batches, it is the agency is expected to have the proper control strategy. That is called step three, changes to the market authorization. So, if you can see the some of the timelines I have given here, both for the EMA and FDA. So, FDA has recently updated the guidance, uh, specifically focusing on the NDSRIs, which can be potentially present in the drug products. So the timelines given for the risk evaluation or risk assessment is 1st November 2023, and for the confirmatory testing is 1st August 2025. That is another 2 years. That is both for the step 2 and step 3. So you can see here, I think a lot of regulatory guidances has come thanks to all the regulators, both FDA, Europe. Europe is quite active. I think a lot of very good guidance has come from the Europe, followed by FDA and other regulators like Brazil, NVISA, and USP has published some in General Chapter 1469. And you can see this is a very crisp slide. I can share it to all the team. You can see very well, this slide, and you can see all the attachments of the recent guidances. And as I mentioned in this slide, you can see recently in July 7th and July 28th, EMA, European Medicines Agency, has made significant important changes in the guidance, which I will talk in the subsequent slides. Then, if you can see this slide, as I mentioned, FDA has recently published a guidance on the acceptable intake limits for NDSRIs, recommended acceptable intake limits for NDSRI guidance for the industry. This was published on 4th August 2023. For the reference, I have attached a guidance here in the PDF. And NPRA, National Pharmaceutical Regulatory Authority, Malaysia, has also published a brief guidance on in June 2023. So what are the, actually this is the one slide talks about, when we start the risk assessment, it is important to understand what are the root causes for the formation and presence of the nitrosamine impurities. You can see on my left hand side the DS, and the right hand side the drug product, the drug substance API and drug product. On my right hand side, you can see the drug substance. These nitrosamine impurities can come from, we need to identify the risk from the route of synthesis. Then lot of API manufacturers will be buying the materials from key starting materials or intermediates from the several contract vendors, contract manufacturing sites. So it is also the responsibility of the API manufacturer, when they buy the materials from outside, they have to do the proper risk assessment with respect to the risk of any nitrosamine impurities or NDSRIs. Then the whatever the solvents, reagents used in the process, because in the API process, people use a lot of recovered solvents to have the cost optimization of the process. So, for example, if in the API process, if a manufacturer using the recycling of dimethyl formamide, so there is a possibility of there is a residual possibility of having the residual dimethylamine in the during the recycling. So that secondary amine itself can give the nitrosamine impurity if it is having any reaction with nitrite or nitrous acids, in the presence of lower pH, it can trigger the nitrosamine impurity. Then packaging materials very important, and cross contamination. Recently, FDA is also very keen about when you are making the same product in uh, uh, different products in the same manufacturing line, how can you control the carry of nitrogen impurities? So how effective your cleaning process? So that is also the recent concern of the agencies. So if you can see on my right hand side, the drug product process, when we have want to evaluate the any possibility risk assessment of the nitrogen impurities in drug product, we will do the assessment with respect to the API, then the drug product process, manufacturing process, and any potential degradation these impurities formation is possible. Then excipients, because the nitrates can potentially come from the excipients. Excipients are the key sources of nitrates, and that can have a reaction with secondary amine under the acidic conditions, or the higher temperatures can potentially give the nitrosamine impurities. And cross contamination, I have already talked about it. And the packaging materials, last but not least, in the blisters, when we use a lot of blisters in the manufacturing of the drug product. So when we use the blisters contain nitrosocellulosic materials, and we always, after the blister packing, there will be a we use the printing of label, like what is the strength, what is the manufacturing date, expiry date. So the printing ink contains, all the printing inks contain secondary amines. So if there is any leaching of the ink into the blister, so it has a reaction with the nitrosocellulosic material, and it can give some type of a nitrosamine impurities. So the evaluation with respect to packaging material is also very critical. So this is with respect to the risk assessment, both from the API and drug product point of view. Then this is the slide, which can give very short summary of what are the potential sources you can see on the table, and like you can see solvents, water, excipients, API, manufacturing process, drug product manufacturing process, and and including the drug product stability. Last but not least, the container closure system. Here you can see, if you can see this slide, there are about 7 simple nitrosamines. All people are well known about this because all the guidance, the first guidance has come somewhere in 2020, September 2021, initial 2021 year. We have about, if you see the FDA guidance, the first guidance of FDA, they have clearly given when you are doing the risk assessment of your API or the drug product, you have to focus on these 7 nitrosamine impurities. These are the simple nitrosamine impurities: NDMA, NDEA, NDBA, etc. But now there is a shift in the regulatory focus. So people have really worked hard, all the industry for last few years, and worked very effectively on the simple nitrosamines. If you can see this, there was a publication of Journal of Pharmaceutical Sciences, which is published in November 2022. The landscape of potential small and drug substance related nitrosamine impurities in pharmaceuticals. Excellent publication. In this publication, uh, if you can see most of the APIs which are there in the market right now, there is about 41% of the APIs and 30% of the API impurities listed in the global database, drug substance database. They are having potential nitrosamine precursors. It means that they have potentially within the structure itself, they have the secondary amine or tertiary amine is already there in the structure. And the problem is when 40% of the APIs and 30% of the API impurities are having the secondary amine within the structure of the molecule. And we are buying a lot of excipients, drug product manufacturer buy a lot of excipients from the market. Then the excipients will invariably contains the small level of nitrates. So the secondary amine present in the API and nitrate under the acidic conditions during the drug product manufacturing or at a higher temperature can potentially form the API related nitrosamine impurities, that is called NDSRIs. And one of the challenge, I'll talk in the next slides, the getting the NDSRI impurities are very difficult. Today, if you see, the availability is not less than, I can say, 5% in the market. Then you can see, based on the literature, you can see number of all the drugs which contains secondary amine group that can react with the traces of nitrate present in solvent, reagent, catalyst, resulting in the formation of NDSRIs. You can see host of structures. I have given only few structures, but you can see many. Everywhere you can see N-nitroso mirabegron, N-nitroso vildagliptin. You can see, wherever the NH group is there, it is replaced with N-nitroso. You can see N-nitroso hydrochlorothiazide. So here, I know that many companies, generic companies, big companies are getting a lot of queries from the European market, from the Germany and Europe, and how, earlier, when industry used to get, generic company used to get the, deficiency from the agency with respect to NDSRI, then they used to work on that and they used to reply. Now, having known that, if there is a secondary amine in the core structure of the API molecule, and if there is a nitrate, present in the excipients invariably, and if your process contains any lower acid conditions or, the higher temperature, it can definitely give impurity. So industry is now proactively working on the, some of the mitigation plans, already started working. Then, as I mentioned quickly on the guidance on the nitrosamine impurities, NPRA has issued guidance in 2023. So, if you can see the guidance, I just put a high level takeaways from the guidance. So, basically, the scope of the guidance is applied for all medicinal product for human use and APIs also, including the biological products. And actually, in the Malaysia guideline, which is published in June 2023, they have not given any timelines like FDA or European Medicines Agency for step 1 and step 2. That means for the risk assessment and for the confirmatory testing. They are not very clear on the timelines. But what the expectation from the regulatory authority of Malaysia is, if you have already done some the risk assessment for the Europe market or FDA, you can share the same documents, for the evaluation to, product registration holder can share the documents with the Malaysian authority. And what is the expectation is for the registered products which are there in the market, if you do the risk assessment now, based on the US FDA guidance or EMA guidance, they are encouraging to do that. And if you get any risk out of the step 1, you need not immediately, report to NPRA Malaysia. But you need to still work on the step 2 and understand what are the level of the identified impurities in the step 1. But for the new product and products under evaluation, definitely the agency is expecting to share the outcome of step 1. That is, if you have any risks in the new product, they are expecting you to present the risks. Then they have also given about what is the expectation of the analytical method sensitivity, what is the limit of quantification of the method. So the guidance is very pretty similar to the expectation of the European Medicines Agency. That is, I'll talk in the subsequent slide for better clarity. But the expectation is, if quantitative testing, you need to, if you have identified the risk, you need to develop a method, and your method should be, LOQ of the method should be 10% of the acceptable limit based on the AI. So I'll talk, what is, for the better clarity, I'll talk about this in the subsequent slides. So basically, NPRA is saying, because you cannot do all the products at a time, so they are telling that you have to prioritize the work, considering the therapeutic indication, treatment duration, maximum daily dose, and number of patients treated. So they have given some, priorities where the PRA, product registration holder, can start working on this. So very, very important, the NPRA expectation is not only to test NDMA and NDEA, because many companies, generic companies, what they are doing, simply during the risk assessment, they are saying that I have done the risk assessment with respect to the formation of NDMA and NDEA, there is no risk, so I am safe. So they are expecting it is not only NDMA and NDEA, you have to do the comprehensive risk assessment, based on the type of the product. Basically, it means that what is the chemical structure, what is the synthetic route. So they are expecting the very comprehensive and holistical review with respect to the nitrosamine assessment. So they have simply, in the guidance, they have clearly given references. The PRH, the private product registration holder, can review USFDA, EMA guidances as a basis. And now briefly, because Malaysia, when you want to do your products, now the risk assessment or the conformity testing followed by the control strategy. So you can definitely use the references as accepted by NPRA. You can use the guidances from the EMA. So I'm going to very briefly touch on the recent guidance. What are the updates in the EMA guidance in the revision 16? Which is published on 7th July, which is very, very important. So earlier. I think, as I mentioned, to arrive at the specification limit, the acceptable intake value is very important. So far, what happened? All the regulators has published the acceptable intake for few nitrosamines only. But from the R&D chemistry assessment, there are so many number of nitrosamine impurities formation is possible. So it is very, very difficult to have the AA value for every nitrosamine impurity, especially for the new nitrosamine impurities, which is not published in the any guidances or not available as per the ICH-M7. So in those cases, earlier European Medicines Agency said, if it is a new nitrosamine impurity, you have to consider the default value of 18 nanogram per day, and FDA says that you have to use 26.5 nanogram per day. But now they have come out with a very good, theoretical concept in 7th July 2023 guidance, that is in the last month. It is based on the carcinogenic potency categorization approach, CPCA. And also they have given in Annexure 3 enhanced aim test conditions for nitrosamines. I'll talk very briefly about what is CPCA, carcinogenic potency categorization approach. So what is the categorization approach? Basically, the document of EMA describes the approach for assigning the N-nitrosamine impurity or NDSRIs to predicted carcinogenic potency category with corresponding acceptable intake limit based on an assessment of activating or deactivating structural features present in the molecule. So basically what they have done, they have come out with a very novel concept. You take a structure, you do the assessment of the structure. So they have defined very clearly in the structure what are the groups that can activating groups, what are the groups which will have a deactivating features, and which are associated with an increasing or decreasing risk of carcinogenic potency. Then basically all this approach assumes the alpha hydroxylation mechanism of the metabolic activation is responsible for mutagenic and highly potent carcinogenic response observed for many nitrosamine impurities. So this is the new concept they have come out, because it is very, very difficult to establish the AA values for all nitrosamine impurities. It is impossible. And the practical challenge what industry is getting, if my nitrosamine impurity is new, I am applying a default value of 18 nanogram per day, and if I maximum daily dose is very high, then I'll get a specification limit is very, very low. So if I get a specification limit of 0.01 ppm, it is very, very difficult for me to develop a sensitive method at such a low level. So industry has really struggled in these years. So thanks to EMA and FDA for giving this such a guidance. It is very, very, this guidance is very helpful. And this CPC approach is applies to the nitrosamines bearing a carbon atom on both sides of the N-nitroso group. You can see the alpha carbon and beta carbons have marked here. So I request, and I recommend strongly to read this guidance. It will be very helpful. Just to summarize the guidance, what the EMA has done, they have given, based on the CPCA approach, they have proposed the AA limits for nitrosamine impurities broadly. They have done the classification. So you can see on my left-hand side, it is on the potency category, where they have come out with a CPCA score, what they call carcinogenic potency score. It is 1, 2, 3, 4, 5. If the score is high, the recommended AA limit is nanogram per day is high. You can see the lowest, potency score is 1, that is 18 nanogram per day. Basically, the N-nitrosamines assigned in this category 1 are predicted to have high carcinogenic potency. And then they have come out with the second category for NDMA and NNK and the the structures which are very close to the NDMA and NNK, these type of things, they have assigned the predicted carcinogen as 100 nanogram per day as a acceptable intake value. Then you can see category 3 they have given as 400, category 4 and 5 they have given as 1500 nanogram. So if it is category 5, your A value is high. If your MDD is 100, if your A value is 1500, if your MD maximum daily dose of the drug product is 100 milligram, 1500 divided by 100, that is, it comes to 150 ppm. So the limit is quite high. So the method development is, so there are so many, it is very convenient actually. So the expectation from the EMA is use this concept. If you have a new nitrosamine, if you have any new nitrosamine impurities, take your API structure, do assessment with respect to this CPC assessment you have to do, and you have to see what AI you have to apply, whether you have to apply 18 or 100 or 400 or 1500. So that will come from the basically from the assessment of the structure with respect to the activating and deactivating groups. So as I mentioned, for you can see here in this slide, the potency score is the alpha hydrogen score plus deactivating feature score, sum of all scores for the features present in the N-nitrosamine activating feature scores. So this guidance is very, very helpful for the industry. And what I understand is there are certain softwares are available to do the CPCA scoring for the molecule. And just to highlight on the importance of the revision 17 of the EMA guidance. So before this guidance of revision 16 and 17, before July, they said if you have a new nitrosamine impurity 18 nanogram per day, you have to apply. And they also given certain cases where the marketing authorization holder is struggling with the low limits. They have also given the leverage of using the temporary acceptable intake of 178 nanogram per day. But now, with the new guidance has come in place, they have removed the concept of this 178. So people are not supposed to use this. This is now they have removed this concept of using 178 nanogram per day for the new nitrosamine impurities. But now they have made one change in revision 16 to revision 17. The treatment duration, if the treatment is up to 12 months of the drug product to the patient, and if the treatment is more than 12 months up to 10 years, they have given certain how to arrive at on the interim limits. They have given certain factors, 13.3 into acceptable intake or 6.7 into acceptable intake. Now what they have done, they have removed the up to 10 years. They have given in the revision 17 duration of the treatment up to 12 months and more than 12 months. They have clearly given that with the factors of 13.3 acceptable intake and 6.7 acceptable. I'll just brief about how to interpret this guidance. Okay, in the next slide. So how to interpret this guidance from the regulator? I just share one example. So here there is a drug product capsule. I have not labeled the product name. And if you apply the guidance, the AA limit for this particular drug product is 100 nanogram per day. This is a published value in the guidance as per ICH M7. And when marketing authorization holder has done the confirmatory testing with the validated method, he found that the limits are out of the limit. Like for example, you can see here the N-nitroso impurity in the drug product impurity, the acceptable intake is 100 nanogram per day, and maximum daily dose is 120 milligram per day. So it is 0.83 ppm is the spec. But when they have tested the batches, it is 1.2 ppm. Then what happened? We have to recall the batch from the market? What to be done? Have we have to initiate the recall? The answer is no, because the guidance, now we have the guidance of revision 17. So there is an interim limit approach they have given. So what is the interim limit? Up to 12 months, I can use 13.3 into AA value factor. If it is the drug product dosage is more than 12 months, I can use 6.7 into AA. So if this particular drug product has comes under the category of more than 12 months, so the interim limit is now, if you can see, it will be the AA value I mentioned in my previous slide as 100 nanogram. So 6.7 nanogram into 100 nanogram is 670 nanogram per day. So the interim spec limit is 670, becomes 670 divided by 120, that is 5.5 ppm. So in my earlier slide, if you can see, it is 1.2, 0.83 ppm limit. Now it has become 5.58 ppm, and my batch is meeting the requirement. But the condition what the agency has put is this interim limit you can use up to only 3 years. In this 3 years period, the marketing authorization holder is expected to correct the process, and and they have to do some risk mitigation to control the impurity to well below 0.83 ppm. I hope this example is very helpful to you for a better understanding. Then, as I mentioned, in 4th August, US FDA has published a very important guidance, which is with immediate implementation with immediate effect. So, if you can see in this guidance, so as I mentioned already, recommended acceptable intake limits for the nitrosamine drug substance related impurities guidance for industry has been published and released on 4th August. And actually, if you see this guidance, it is based on the European guidance released in July, 7th July and 28th July, and also Health Canada has given certain guidance. So this is also very similar to that one. But the only thing, the difference in the EMA guidance and FDA guidance is for the category 1, EMA guidance is set 18 nanogram, you have to consider as a AA value. If the CPCA score is for the category 1, FDA says to consider 26.5 nanogram per day. And also FDA has published the AA values, acceptable intake values for the almost 247 nitrosamine drug substance related impurities. And whereas the EMA has given for about 83 nitrosamine drug substance related impurities. So put together the guidance approximately about 347, 350 NDSRIs, there is a AA value is published. So you can see this table. You can see, if you can see this table, it is quite similar to the EMA, except the limit for the recommended limit for the A in nanogram per day for the category 1 potency. It is just the 26.5 nanogram per day, but against the 18 nanogram per day suggested by EMA. So what the guidance is saying, the guidance again is applicable to all the OTC products. The applications are pending. That basically the FDA expectation is you have to re-evaluate all your products. If your drug products containing the drug substance contain secondary amine, you have to do the re-evaluation of your risk with respect to the any potential formation of NDSRIs. And you can apply the AA values published in the annexure of the guidance. That what I said, 247 molecule for the NDSRIs, they have published the AA values. If you have a new NDSRI which is not there in the list, you have to arrive at a AA value based on the by applying the CPCA concept. So that is the expectation. And so that they have also given timelines, as I mentioned earlier. The risk assessment, the re-evaluation of the risk with respect to NDSRIs present in the drug products, the risk assessment is only 3 months. We are already in the mid of August, so September, October, November 1st, the risk assessment need to be presented. And however, once you identify the risk for developing the method, validating the method, and testing the batches, and correcting the process, they have given the timelines almost for the 2 years. That is August 1st, 2025. But FDA has made a very clear expectation. If any drug product batch found contains levels of NDSRI above the FDA recommended limit based on the AA, it should not be released to market by the drug product manufacturer, and they have to remove from the market. And they are also encouraging FDA. The manufacturer should contact the agency if the recall is initiated, and FDA may exercise the enforcement discretion when warranted to prevent the mitigate the shortage of drugs in the market. So they have given really very good clear cut instruction to the clear clarification to the industry. So just these are all the regulations. So what are the general challenges what we face in analytical? So just to update to the team, because all the companies are already started working on the nitrosamines from last 3 years. So the Definitely, they require a highly sensitive and selective methods which can use both LC-MS-MS and GC-MS-MS, because HPLC or conventional GC HPLC, the sensitivity to detect at a low levels. Like for example, FDA expectation is to have 0.03 ppm of LOQ, which it is very difficult to achieve in the HPLC. So high techniques are required, and when we develop the method, it should be selective, sensitive, with good accuracy and precision, and we need to have a good detection factor. And USP 1416 are also given some general procedures, but please note that no regulator has given any procedure officially in the any monograph. So these are any general procedures given, but the general procedures can be, definitely applied in the lab, and we have to fine tune the method with respect to the particular API or the drug product. And this is a very important slide. How do we establish the limit? So once we establish the limit, you know how to establish the limit. I have mentioned in my previous slide. The specification limit for any nitrosamine impurity is established by A value in nanogram per day divided by maximum daily dose in milligram per day. So in the specification limit is for the nitrosamine impurities, for example, 1 ppm arrived based on the formula. The expected LOQ is 0.1 ppm. So if you develop the method with a LOQ of 0.1 ppm, that is 10% of the AA, and if you test all the batches of drug product, all the batches in the sense you select some 10 batches, the means freshly manufactured batches, mid of stability, end of stability, and after expiry. You can, if you test all these batches, if you in all the batches your nitrosamine impurity is less than LOQ, that is 0.1 ppm, 10% of the AA, you need not do any further testing. You can skip, you can do the omission, and you can simply skip the testing. But if you have, by any difficulty, if you are not able to develop the method with 0.1 ppm and you have developed the method with 0.3 ppm, 30% of the AA, then the agency's expectation is at least 10% of the annual batches need to be tested. That means if the nitrosamine impurity batches are in between 0.1 to 0.3 ppm. And if any chance, it is, if analyst having any difficulty to get the LOQ even a 0.3 ppm, if they do it 1 ppm equivalent to the specification limit, then 100% of the batches need to be tested. This is the EMA approach. Many industries following, but FDA is saying that whatever you do, you have to have a default LOQ for the nitrosamine impurity limit as 0.03 ppm if the maximum daily dose is less than 1 gram. So what I am trying to say, the importance of the analytical scientist in the whole nitrosamine scenario, it is very important. We the industry should have a very good scientist on the bench who can really work on the sensitive and very good robust method. Then FDA has also given certain expectations, like the how to mitigate these nitrosamines, because if you see lot of all excipients, I have read some literatures. They are very right in a way. In 2011 also, in the AAPS, there were some good publications published. If most of the excipients invariably contains a trace level of nitrates, like anywhere between the 1 ppm to 10 ppm. So if the nitrates are present at such a low level, there is a high possibility of this nitrate which is present in the ppm level can have a reaction with the secondary amine, present in the API or present in the process, that can react under the acidic conditions or under the elevated heat conditions, which can generate the some type of a nitrosamine impurity. So how do we control this? So it is very difficult. So, but FDA has given certain guidance. Basically, one of the simple guidance they are saying, when you are designing your drug product manufacturing, you can try to keep some basic conditions, so that in the basic conditions there is no chance of formation of nitrosamine impurity. You can keep the pH above 5 or above 6, the product pH. And they are also saying that you can use also some scavengers like ascorbic acid, caffeine, phenolic acids, and all these things, alpha tocopherol you can use, But one of the practical challenge for the generic industry is, already the formulation is fixed, the bioavailability is proved. If you any fine tune these formulations by adding these scavengers at certain level, what is the impact of the bioavailability on the formulation, and what is the change in the in vitro dissolution. So all these things need to be studied. So the solution is not very simple. What is given by the regulator, it needs a lot of work to be done by the industry. So just to move on. So I have seen, one interesting presentation given by one of the company in India in Bangalore. The company name is Circutech. In that, there is a how to minimize the possibility of formation of NDSRI impurity during the drug product manufacturing. So they have some, very novel solution. Controlling the nitrogen potential formation of the NDSRIs impurities during the drug product manufacturing by using ionization technology. So they use static controls. They use the ionized air instead of the regular compressed air. So this concept is very novel. Actually, they have working with some bigger companies in India. This company name is Circutech Taco. They are working with bigger companies. They are working with Dr. Reddy's, Cipla, Piramal, Laurus, and all big companies. Milan in India. They have seen some successes. When using this very simple, cost-effective novel technology to and which can really prevent the formation of the NDSRIs formation during the drug product process. So I have given the address here. I can share this presentation with Mopi. If anybody is interested, they can contact Mr. Nanda Kishore from Circutech. Then once we have the drug product risk assessment, I think the expectation is to write I think it is also very important to write the very type of typical risk assessment report, which can have the objective of the report and drug product details, that is the product name, market, strength and all, drug product composition, whether it is a premix, binding, blending, packaging, etc. We have to write about the risk analysis and evaluation of the potential sources. So what is the risk identified from the drug substance manufacturer? What are the risks identified from the excipients? Because the drug product manufacturer is expected to, he has to get all the reports from the suppliers, with respect to the nitrosamine, their risk assessment reports. So based on the risk assessment reports, drug product manufacturer has to do the risk assessment report. The excipients, packaging materials, and the drug product manufacturer will also do their, with respect to the equipment, any possibility of the nitrosamine impurities formation, manufacturing process, then facility, then cleaning agents, because they use sometimes detergents in the killing of the equipment. So if any cleaning agents can possibility to bring the nitrosamine impurities, then purified water, then lot of any confirmatory testing has been done. The data will be included in the drug product report, and basically finally it will be summarized with the outcome of the confirmatory testing and summary before giving all the. This report will be around 30 to 35 pages. Along with the annexures, the report will go up to 200 pages. This will be submitted to the agency. So definitely the report submission requires a lot of due diligence, understanding, and I want to emphasize also Nitrosamine's work is really a collaborative effort. It requires a lot of cross-functional collaboration like R&D, manufacturing, QC, QA, regulatory affairs. Everybody has to work hand to hand to make work on this risk evaluation and confirmatory testing followed by the control strategy. Because really a teamwork is needed. Otherwise, it is not going to be very successful. So what are the conclusions and way forward? So product manufacturer should know the product thoroughly, and you need to have a constant communication with the API manufacturer, whether API can give any risk of nitrosamines or NDSRIs, and formulation need to be understand. Sometimes the excipients also contains some potentially formed to N-nitrosamine impurities, like trolamine. It has secondary amine group, the excipient. So the secondary amine group can be replaced with NO. It can form potential nitrosamine impurities. Okay, some FNDC colors, preservatives may also be the potential source of nitrosamine impurities or nitrosating agents. That we need to have a very clear understanding that the manufacturing process, whether the process involves low pH or higher temperatures conditions, all these things need to be studied very carefully. And when the NDSRI is possible, the test excipients for nitrates, and it need to work with the experts, to control the level of nitrates present in the excipients. So from the December 2021, I have made 4 publications. That is my first publication on nitrosamine impurities current regulatory status in December 21, followed by July 2022 on N-nitroso impurities, then February 23 and July 23, and I'm also publishing on the recent guidance on September 1st, 2023. My 5th publication is coming. These were published in a magazine called Cutting Edge, published by Spinco Biotech. You can see online. They're available at a free of cost. I strongly encourage to read these articles. So if you read all these 4 articles and my upcoming article, which I can share on the 1st of September, if you read all the 5 articles, you need not refer any guidances, because all the guidances, what is there, I have summarized nicely in these articles. So I am from a consulting company called Q Dot Associates. So after I quit from my company, Dr. Reddy's, I worked for about 14 years overall. In my last stint, about 7 years at a corporate quality control and quality assurance as a vice president in corporate quality control. So I started working with the consultancy, and we are working with big companies. I am also continuing as a consultant to Dr. Reddy's in India and other big companies in India. And the services what we are offering, this is my brief profile, and this is the profile of my colleague Dr. Satish. And what we are supporting is, we are also supporting some of the clients with respect to the nitrosamine risk assessment, end-to-end risk assessment we are supporting. In addition to, we will be also supporting the review of the investigations, OOS investigations, suggesting that whether we will review whether the CAPAs are effective. We have a team of about 8 people. We are working with international clients. Right now I am doing this webinar from Malaysia, working with one of the clients in Malaysia, and we have expertise in doing the GAMP assessment with respect to the GAMP, and also data integrity assessment, and all the sorry. We also do. We have also team to support the regulatory inspection readiness, and if there are any observations from how to write the response to the agency, and we also do due diligence audits, supplier qualification, vendor audits, and we have our own software tools to support the sample management and also learning management system. And we are also doing a lot of competency building, coaching and trainings with the teams. So thank you for your attention. I hope my session will be useful to you and it will be very helpful. And if you have any doubts or any clarifications needed on nitrosamines or NDSRIs, or the areas where we are working, you may please contact me. My name is Dr. B.M. Rao from Q Dot Associates. My email ID is drbmrao@q.associates.com. Thank you so much.